Welcome to your lesson 2 on meiosis. We're going to be covering Mendel and heredity, and we're just going to follow along with your lesson 2 notes packet that you should have received last class for homework. If you have not gotten this packet, you can access it off of Canvas and follow along with us. So let's start out with knowing what traits are. So traits are distinguishing characteristics that are inherited. A trait is not what color nail polish you have on. That's something you choose. But a trait is something you actually inherit from your parents. So for these cats, they have different fur textures, like a little fluffy one here, a little bald cat there. They have different fur colors, patterns, tail lengths, ear shapes, eye colors. So these are all things that are inherited, and we call them traits. Gregor Mendel is the person that showed how traits are inherited. He showed that they're inherited as discrete units. Many people back then thought traits were blended. And what they mean by this is that if you had a red flower and you had a blue flower and you cross those two things together, you would end up with a purple flower. He showed that actually if you have a red flower, which these are scribbles or flowers, and a blue flower, and you cross them, you could actually get red flowers or blue flowers, right? So they show that the traits are actually inherited discretely. Now he worked with pea plants because he knew that he could control their pollination and that they reproduce quickly. You don't want to plant like a tree that's going to take 100 years to grow between each trial. So these plants grew pretty quickly. So in your notes, it tells you to outline his basic experimental design, which is what we're going to do here. You can see he actually cut a piece of the stamen off, so the pollen, and he would take that pollen and he would pollinate another plant, right? So let's say he took the pollen from the white flower and he pollinated the purple flower. Meanwhile, he'd cut off the stamen of the purple flower. That way it would not self-pollinate, right? So he knows for sure that this is one parent and this is the other parent. And when he crossed these two, the parent that was growing the new pea pod, which has been this purple one, grew seeds. Remember, peas are the seeds of the plant. So then he would take this pea pod and he would replant these individual peas here into the ground. And they would produce offspring. So this was the plant's offspring. And then he would make observations. So he knew the types of the plants that were being crossed. So he knew that he was crossing a purple plant. He was crossing it with a white plant. And then out of all of the offspring, he only had purple plants. So he'd make these observations. He would then go back and then cross some of these purple plants together, or he would have the purple plant self-pollinate here. So he might have the purple plant self-pollinate, so allowing these individual pollen here pollinate the same plant. Okay. So what did he end up observing? So he particularly looked at traits that were discrete, meaning they are not affected by crossing over. These traits are very far apart, probably on different chromosomes. So these traits were inherited completely discretely of each other, which means just because the plant is purple, it's not even a letter, just because the plant was purple does not mean that it's also tall, right? It could be purple and short, it could be tall and white, doesn't matter. So to go over some vocabulary here, a genetic cross is the mating of two organisms. It's just a scientific word for the two organisms had a baby. The P generation is the parent generation. The F1 generation is that offspring. And then F2 is the result of self-pollination of F1. So when we were looking at our last slide, you had the purple flower crossed with the white flower. So that was the P generation. Then you had the 4 offspring here, which were all purple flowers. This was F1. And then if I took this purple flower and crossed it with itself, I would get then the F2 generation, which has yet to be seen. We haven't gone over what that generation would look like. All right, so some of the traits he looked at here, and you have the same image on your paper. You could look at flower color, flower position. So flower colors are purple or white. Flower positions, axial, meaning at the top, or terminal on the sides of the plant. You could have yellow or green seeds, round or wrinkled seeds, inflated or constricted pods, green or yellow pea color, and tall or dwarf for short stem length. So these are called traits here, and these are called alleles, right? So you have the purple allele and the white allele of the flower color trait. Now, what Mendel concluded is that certain traits did not disappear. He concluded that they were just temporarily hidden. For instance, when he crossed that purple plant with itself, he found that some of their offspring were white. So just like they had inherited that white gene from their grandparents. So if you've ever heard of genes skipping a generation, this is what we're talking about. Those traits didn't just disappear all of a sudden, they were just hidden. He concluded that traits are unique factors that are passed from parent to offspring. So going over our chromosome picture here, you can label the picture in your notes. A gene is a segment of DNA that provides instructions for something. So this location is the gene. So this is the locus or location of the gene. And this gene is for flower color. The allele is the version, so purple or white, that's the allele. And remember, these are homologous pairs. That's like a pair of shoes there. They're similar, but not perfectly identical. Right, major vocabulary here you will definitely need to know. Hetero means different. Homo means same. Zygous refers to the allele. So this means the two alleles are different. For instance, you have purple and white are the two alleles for those flower color. It says white, pretend. Homozygous means the alleles are same. So you have two purple flowers, right, on each allele. So in that last picture we looked at, you had the two chromosomes. It's talking about here and here. If these two are the same, it's homozygous. If they are different, it's heterozygous. So you can see here, for this gene, they'd be homozygous. They're both for pink. For this gene, or excuse me, homozygous because they're both pink. For this gene, they're heterozygous. One's orange and one is green. So we have a little bit more vocabulary here. A genotype is the actual genetic makeup. So this is showing on your chromosomes you have a purple or flower, and uppercase letters indicate dominant traits, and a lowercase letter is a recessive trait. So you would say a purple flower and a white flower. We like to use the same letter when we're talking about genes. So you would not use purple and white. That gets too confusing. We want to use the same letter, an uppercase version and a lowercase version. So for instance, P and little p, right? Now, a phenotype is what's actually physically expressed, right? Or what's seen on the surface. Dominant traits are always seen if they are there. So if you have different traits, like for heterozygous traits, this flower, so that's purple and that's white, this flower would be purple, right? So would this flower would be purple, right? So they have the same phenotype, but they have different genotypes. So genotypes are the actual letter combination in the DNA, and phenotype, so I think phenophysical, phenotype is what's seen on the surface, what's actually expressed. So allele combinations are represented by letters. So we're going to go through an example here. So let's say this T, this gene we're looking at, is height. So let's say this is for tall. So tall is going to equal capital T, and short is going to equal lowercase t. So we'll put that over here. Now, for this, so for here, we have our genotype. It's going to be one tall allele and one short. So our alleles are going to be T for tall, little t for short, but our phenotype here, because there is the big T, is going to be tall, right? So we would call this combination heterozygous. Here they are both tall, right? Both uppercase T's, but they're the same. So these are going to be homozygous. Now, in order to know that they're homozygous, but they're both uppercase, we would call this homozygous dominant. Right? So homozygous dominant. Which, when we have two little t's, this is going to be a short plant because there is no dominant gene to overshadow the short gene. And we are going to call these also homozygous. But now it's going to be homozygous recessive. Right? So you can see the only time that the short gene or the recessive gene will show is when there is no dominant gene available. The dominant gene is kind of like that really outspoken friend, that if you have a group of friends together, that really outspoken friend is the one that everyone's going to notice because they're kind of overshadowing everybody else. Somebody who's more quiet, they might not be noticed in a crowd unless, well, everybody else is quiet, and then it changes the perception of other people. So I always think of the dominant friend as that like really over the top person, and the recessive gene as more of somebody who might be a little bit more shy and not be noticed unless there's around other shy people. So all of this is really simple as far as, the dominant trait shows up, the recessive trait does not, and it's very easy. Except that this is not how actually humans inherit traits. You see, most traits do not follow Mendelian patterns or do not follow the patterns that Mendel observed. So very few traits. So we're on exploration 3 now. You should be on page 3 of your notes. So when the question asks how many traits follow the Mendelian patterns, very few, especially for humans. We have much more complex genes than that. So the way we actually inherit genes is we have things called incomplete dominance and codominance. For instance, incomplete dominance, when you crossed a red and a white flower, incomplete dominance would mean you get a pink flower. This is like that blending example we looked at earlier. Codominance means you both get white and red, and that they are showing up as like stripes or dots or things like that. We often see this in the fur color of animals. For instance, if you have a chocolate lab and a white husky, right? You might get a dog that has patches of both chocolate and white fur, which is exactly what my husky lab mix has. So you get that codominance there. Incomplete dominance is more like something if you have a parent with black hair and a parent with blonde hair, you might be born with light brown hair, right? So incomplete dominance is a blend, and codominance equally expresses those traits. Multiple alleles, so we're down to this, is when you have more than one allele. So, or excuse me, more than 2 alleles. So before we've only been looking at like, capital P and lowercase p. But in this case, you could have, for like blood type, you could have IA, you could have IB, or you could have little I. And what this means is that these 2 types are codominant and this type is recessive. So the combinations get a little bit more fuzzy here. And you have, instead of having 2 possibilities of phenotype, remember that's the physical expression, you now have 4 possibilities of phenotype. So you could be blood type A, and that's if you got 2 A's. You could be blood type B, and that's if you got 2 B's. You'd be blood type AB, and that's getting 1 A and 1 B. Or you could be blood type O, and that's getting 2 of the little i's here. So there's a few more activities that we will do with blood types here. If you think about it, basically A and B are co-dominant here. So you either have A, you have B, you have AB, or you have O. And so this is because there are multiple alleles, not just two. All right, finally, we have sex-linked traits. All the traits we've looked so far are going to be on our autosomes. Sex-linked traits are not on the autosomes. They are on the X and Y chromosomes, or our sex chromosomes. But the X chromosome is much larger than the Y chromosome. So the Y chromosome is a much smaller chromosome. So your locations, remember on the autosomes up here, the locations of the genes were at the same place. They can't be at the same place on X and Y because they're two different chromosomes. So females have 2 copies of X chromosomes, and males have only 1 copy of the X chromosome. So for females, if they have two chances for every trait, and males only have 1 chance, right? So you can see here, a woman who's heterozygous for this trait, see a capital H and a little h, they can still be normal. Whereas a male, they only have 1 trait. So if they get that 1 trait that's recessive, they have the phenotype. So we can see this with hemophilia. Hemophilia meaning your blood does not clot. So in order to have normal blood, you have to have the dominant trait, right? So males with the dominant trait on the X chromosome will have normal blood. Females with at least 1 dominant trait will still be normal. They will only have hemophilia if they have the recessive trait on both X chromosomes. Because males only have 1 X chromosome, they only get 1 chance. I think of sex-linked traits like this. If I gave you all a quiz, and then I said, okay, I'm going to take the highest of your 2 grades, but only the girls get to take it again. The boys only get the 1 chance. Doesn't seem very fair, obviously, but genes don't care about fairness, and that's how sex-linked traits work. And so this is why we often see sex-linked traits are more common in males than female. So if it's a sex-linked trait, we're more commonly going to see it in the male population. It can still happen in females, but it's much more common to happen in males. This is things like color blindness and hemophilia are 2 examples you're going to see most often. All right. Finally, we have polygenic traits. Polygenic traits can be produced by 2 or more genes, which means you have a chromosome and this gene codes for it, and over on this chromosome, this gene codes for it, and this chromosome, this gene codes for it. So you have multiple traits interacting. This is going to be most of your traits. Like eye color has like 15 different genes that control it. Height has like 40 something. There's all, 600 genes affect your height. Excuse me. So each trait here has a lot of interactions from a lot of genes. These are not easy to figure out. And so what the result of this though is for polygenic traits, you end up with like a bell curve here, meaning you don't either have either or. It's not going to be a bar graph. You're going to have some people that are extremely short, some people that are extremely tall, and then it's going to be a range to where this is your average height, right? You're not either 5 foot or 6 foot. You could be anywhere in between. You don't have brown eyes or blue eyes. You could have any shade in between that. So most of your traits are considered polygenic. And so these are going to be traits that are produced by more 2 genes. Okay, almost done. Epistasis is basically a gene that suppresses another gene. So for instance, you could have a gene for blonde hair, you could have a gene for red hair, but if you have the gene for baldness, it doesn't matter what color hair you have because you're going to be bald. Right? We see this a lot in dogs with certain fur colors. So really blonde dogs tend to have a gene to be blonde across the board. And so they might have the gene for chocolate fur, but it doesn't matter if they have that blonde gene there. So this is epistasis, basically suppresses another gene. So the baldness gene, doesn't matter what color gene color hair you have, the baldness gene overrides it. The last thing I want to mention here is that the environment interacts with genes by changing the way they are expressed in a phenotype. So you have your genes, what you're born with. Let's say those 600 genes that code for your height say that you're going to be on average pretty tall. But even if those genes say that you're going to be pretty tall, you have to take into account environmental factors. For instance, if you eat a bunch of junk food your whole life as you're growing, or you don't get enough nutrition, from eating that junk food, you might grow shorter than your genes had originally planned. Other examples are skin tone. If you spend a lot of time in the sun, your skin tone is going to be slightly darker than if you didn't. For a plant environment, all three of these plants could have these same genes, but maybe this one got more water and sunlight, and this one got less water and sunlight. So we can change the expression of the genes, or what is expressed physically, by changing the environment. And this is why scientists push so often for clean air and healthy eating and drinking enough water, because we want our genes to be able to express themselves fully. So that is the end of lesson 2 here. If you would like to rewatch the video, it is on Canvas. So you can go back through your notes, pause the video throughout. And if you have any questions, remember to message me on Remind, or watch the other Amoeba Sisters videos as well that are up here about these same topics. Right? Thank you.